Hello everyone, my name is Alexandria Quinlan. Thank you for joining me today as I discuss my project, Ferns on Ferns, an exploration of epiphytic fern gametophytes growing on low trunks of tree ferns in Taiwan. This poster summarizes sections of my master's work advised by Dr. Liuong Guo, who you can find at the conference. However, since I am not present at IBC, I wanted to offer a virtual presentation that you can follow while viewing the poster or watch later on. To begin, this project was largely inspired by studies which have explored epiphytes growing on tree ferns being species that belong to Cyatheaceae, Dixoniaceae, and Blecknaceae. These studies found that tree ferns are substantial hosts for other plants, often composed of distinct epiphytic communities that are considerably different than those on surrounding hosts. Particularly, certain epiphytic fern species have been observed exclusively growing on tree ferns, presenting an extreme case of host specificity that is predominantly found in fern epiphytes. However, this fern on tree fern epiphytism has yet to be substantially explored for the gametophyte generation of ferns. Ferns are unique in that they have two independently existing generations, the haploid gametophyte and the diploid sporophyte, where the gametophyte has many responsibilities, including sexual reproduction, habitat selection, and migration. However, gametophytes are often overlooked due to their obscure morphology and small size. If you look at these photos here, you can see the small size that I am referencing with a corniform gametophyte on the tree fern root mantle, and a zoom in of one of the gametophytes from our study. As fern research further investigates this generation, we're gaining more insight into the ecology, physiology, and distribution of different fern species. This study in particular aimed to determine what factors make tree ferns optimal habitats for gametophytes and identify differences in fern gametophyte communities between tree ferns and other hosts. Our study was conducted in Wulai, Taiwan, a small town outside of Taipei that is home to many subtropical forests with a year-long rainy season. Here we established 6 circular plots, 3 surveyed in June of 2021 and 3 surveyed in October, that were each 5 meters in diameter with an Alsophyllus spinulosa host at its center. Criteria for plot selection included finding tree fern hosts with an epiphytic community established, which also had angiosperm hosts growing nearby for comparison. All fern gametophytes beneath 1 meter on host trunks were collected. A garden net, which you can see here, was used to generate subplots to assist with collection. Additionally, in the field, we installed hobo loggers on 1 live tree fern, 1 dead tree fern, and 1 angiosperm trunk to investigate the relative humidity of the hosts. Back in the lab, after all samples were cleaned and photographed, they were identified to species level. We ended up processing over 150 samples for June and 240 samples for October. We followed a tissue-direct PCR protocol, a process which bypasses DNA extraction, where you take a microscopic piece of gametophyte tissue and break it down to be used as the DNA template. Amplicons were then pulled into a library and sent off to NGS for sequencing. From there, we followed this bioinformatics pipeline to clean sequences and blast them against a local library and the NCBI gene bank. This pipeline here also includes a section on primer design, and this entire pipeline is currently in review for publication. Between the 2 surveys, there was a total of 32 gametophyte species identified, where the average species richness was around 8 for tree fern trunks and around 3 for angiosperm trunks. The NMDS here, which is figure 1 on the poster, illustrates species composition both by host type, with angiosperms shaded in orange and tree ferns shaded in blue, and by season, with ellipses distinguishing either group. A permanova was performed which found that species compositions were significantly different both between host types and seasons. The names for these abbreviations are provided in Table 1, as well as information on morphotype and presence on the hosts. I'll discuss morphotypes more in a later slide, but for now I'll mention that the two types are cordiform and non-cordiform. This is generalized as there are some species, such as Angiopteris lygodifolia and Lemophyllum microphyllum right here, which do not strictly fall into either category. The bold species, both in the NMDS and in the table, represent those species which have an accidentally epiphytic lifestyle, meaning that these species would normally be found growing terrestrially. You can observe from the NMDS that most of these species were found growing on the tree fern host type. A final thing to note here are the asterisks next to a few species, these small asterisks here, here, and here. And these asterisks are representative of species which have no conspecific sporophytes growing in the study region. Callicopteris apifolia and Haploteris eucusiamensis were confirmed as independent gametophytes, which you can observe in Figure 5, and I will discuss more in the slides to come, while Antrophium henryi and Haploteris flexuosa had record of sporophytes in nearby regions. We'll now take a look at Figure 2 on the poster. With a difference in sample sizes, that being 7 hosts for tree ferns and 16 hosts for angiosperms, we chose to analyze species richness with a species accumulation curve, which revealed that as surface area of the hosts increase, cumulative species richness was higher in tree ferns. A generalized linear model was also performed, which is here on the right, to investigate the effects that surface area and host type have on abundance. For angiosperms, as surface area increased, so did abundance of gametophyte species growing on the trunk. This pattern, however, was not observed in tree ferns, where you can see that the tree fern with the highest abundance of just over 75 gametophytes on the trunk had about a median surface area. Nevertheless, both surface area and host type were found to be significant predictors of abundance, with tree ferns hosting a higher count of fern gametophytes. In addition to species composition, we observed differences in ecomorphology of gametophytes on hosts. Fern gametophytes are further diversified by type, where a cordiform, short-lived morphotype is generally associated with terrestrial growth, and a non-cordiform, long-lived morphotype is generally associated with epiphytism. The ribbon, strap, and filamentous morphotypes have adapted a branching of the gametophyte thallus, with some species producing gemma, another adaptation I'll mention more in the next slide. For Figure 3 here, the top box plots illustrate the density of morphotypes on each host type, with individuals per meters squared on the left and species per meters squared on the right. The bottom set is for lifestyles, where epiphytic is referring to obligate epiphytes, accidental is referring to species whose habitats are normally terrestrial, and facultative are species which can adapt to either habitat. Tree ferns primarily hosted cordiform gametophytes, which were accidentally epiphytic, while angiosperms more frequently hosted non-cordiform epiphytic gametophytes. A final important result from species composition is the presence of independent gametophytes, which are species that have no conspecific sporophyte growing nearby. Both of these species have been reported before as exhibiting an independent lifestyle. Haploteris eucusiamensis was quite abundant in this study, growing prolifically on both host types. This species exhibits an extreme case of independence, where a genetically identical sporophyte has yet to be found in East Asia. For the Callistoteris species, only 2 individuals were found in the study region, one growing on an angiosperm host and one on a different tree fern species, also Phyllopodophylla, outside of the plotting region. Since the Apodophylla hosts were outside of the plotting region, their data were not included in the analyses. However, we were still able to identify some of the gametophyte species growing on their trunks. As mentioned, independent fern gametophytes often utilize gemma for apogamous reproduction, which can be seen in both of these species here with these gemma and here, and is likely why they were able to persist in our study region without a sporophyte nearby. Gemma are small vegetative propagules that disperse and can give rise to a new yet clonal population. The final result I'll touch on is relative humidity, illustrated in Figure 6 on the poster. While loggers were installed for the duration of the study, only the data from May to August was analyzed due to inconsistent recording. As mentioned, our study site was in a forest with a year-long rainy season, yielding high humidity results for all 3 hosts. All 3 of the recorded hosts spent most of their time at values above 95% relative humidity. However, an ANOVA followed by a Tukey post hoc test found that the live tree fern and the dead tree fern spent significantly less time under 85% RH than the angiosperm host, dropping below the threshold and staying there for a shorter period of time. A drop threshold of 85% was informed by a different master's project on fern gametophytes, also advised by Liu and Guo, which established that RH values below 85 are not suitable for desiccation-intolerant fern gametophytes. The figure on the right shows the duration that hosts spent across vapor pressure deficit ranges. All hosts spent substantial time in ranges below 0.25 kilopascals, with the tree ferns remaining at 0 for most of the study. These low VPD values are further indicative of the high humidity levels at our sites. While there is much that can be discussed here, I'll only be touching on 3 main points. Firstly, the high abundance and species richness of fern gametophytes on tree ferns is likely associated with the stable relative humidity of the moist root mantle, as well as substrate porosity. In 2005, Meltretter et al. Conducted a study which explored epiphytic compositions of tree ferns in Mexico and found that tree ferns have a higher water retention capacity than angiosperm hosts. Humidity and moisture are especially important for fern gametophytes for protection of their single-cell layer tissue and for the necessity of water for their motile sperm. Further, the tree ferns in this study were a substantial host for accidentally epiphytic gametophytes. While some gametophytes have been known to exhibit desiccation tolerance, gametophytes of terrestrial fern species often have low DT and a preference for moist, protected sites, which the tree ferns here were likely able to provide. Additionally, as humus accumulates on the sponge-like root mantle, the tree ferns simulate a terrestrial environment. These characteristics allow tree ferns to support plants without a strictly epiphytic lifestyle. The final point I'll touch on is that the differences revealed in fern, sporophyte, and gametophyte species compositions in this study, as well as the presence of 2 independent gametophyte species, highlights the importance of studying the gametophyte generation. We'd like to thank Dr. Liuang Guo's lab at National Tsinghua University, Dr. Zhiming Hu's lab at National Taiwan University, the Taiwan Forestry Research Institute, Dr. Zeleny for his assistance with community ecology, all of the students who assisted with the survey, and most importantly, we would like to thank the tree ferns of Neidong Dao. As mentioned, this poster summarizes a few sections of my master's thesis, but I would love to share more. If you'd like to chat about ferns on ferns, or flatworms on tree ferns like this friend here, or other spore-producing organisms, you can email me, or you can find me on social media at Fervor for Ferns. Thank you for your time, and I hope you've enjoyed my poster.